If you would, turn to 10.1. We're going to review what we did yesterday here, talking about meiosis. Again, this is your essay. If you get chapter 10.
Probably don't have a quiz.
There's no quiz today. Really, all the steps of meiosis are there for you on page 273. If you want to turn to 273, that's a good place to look to try and understand what we're doing here. Okay, hold on, let me get this before we get going. Andrew Mullis is not here. Graham is not here.
He was working with Bennett.
And Jay in the computer lab.
And then Jay is not here. And Bennett.
Yeah, he was working with Andrew.
Alright. Here's a cell. The cell is located in the testes or ovaries. It's going to create your sex cells, your gametes. Okay?
Good.
First step, we go through the phases. Prophase, metaphase, anaphase, telophase. We go through them twice.
There they are.
Prophase, metaphase, anaphase, telophase, cytokinesis.
Those aren't working here.
Yep, you got to figure that out
That's
on your own. We're on page 273. First thing that happens, prophase 1, nuclear envelope disappears.
Centrioles go to outside.
Centrioles go to opposite sides.
Chromatin tighten.
Spindle fibers come out. I'm not going to draw those. We can deal with that.
Chromatin tighten.
But when we're making sex cells, it's a little bit different. Oh yeah, chromosomes coil up so you can see them.
Nucleolus disappears.
Nucleolus disappears.
This crossing over.
The homologous.
No, they have to find
The
that.
mounting of the cell.
Thank you.
Where is that box? Because it should be. Okay.
The homologous
What?
chromosomes go together. Cells not
The homologous chromosomes will come together and exchange. Homologous chromosomes come together.
What's
Homologous
that
chromosomes come
called? What's
together.
that called when I find that pair?
This is called a bivalent. It's also called a tetrad.
We didn't know that.
It's also called a homologous pair. I'm not sure which was the one they used in your reading. Yeah, you can just call them homologous pairs. They have 3 names. Bivalent, tetrad, or homologous pair. And anyway, those come together and exchange genetic information. That's called crossing over. That's our first source of variation. Why do we want variation? Graham?
I thought I knew, but.
Why do we want variation?
So not every cell is the same.
So you can have all the sperm and egg cells different. I'll talk more about that in a minute. But you never get the same, the kids never look like the parents and they never look like each other. And this is one of the sources of variation crossing over. Now we have a chromosome that's different. Metaphase 1, the pairs line up in the middle.
But it's different because they line up in pairs.
Notice every time this happens, they don't have to line up the same way. The next time this happens, they might line up like that, with 2 whites on the right. Or they could line up like this, with 2 greens on the right. Or they could line up like this, with 3 greens on the right. Yes.
Does those be the same size with each other?
Yes, the same size always comes together. Those are called homologous pairs.
And centromeres.
And what?
Like the centromeres have to be the same.
Yeah, they do. They do.
When do they mount each other? Like, get on top of each other?
That's prophase 1.
That's crossing over.
That's crossing over. We already did that.
That's what I thought.
But we call this, the way they line up, is called independent assortment. That's an important source of variation. They independently line up randomly. Each pair lines up randomly in metaphase. So we call that independent assortment. That's our second form of variation. Then these things are going to separate in anaphase and go to opposite sides. And in telophase, we form new nuclei on either side. Marker.
Right behind the board. Wait. Behind the figures.
New nucleus forms. New nucleus forms. Cell pinches in the middle.
Furrow.
Yeah, that's called a cell furrow. And now we have two cells. What's different about these cells than at the start? Major difference.
What about 3 chromosomes instead of 6?
They only have 3 instead of 6, and what do we call that?
Haploid.
Haploid. These cells are haploid. The first cell we started with had 2 sets of chromosomes. This cell only has 1 set of chromosomes. It only has 1 of the big one, 1 of the medium one, and 1 of the small one. The first set we had 2 of the big one, 2 of the medium, and 2 of the small. So this set has 1 set of chromosomes. The old cell had 2 sets of chromosomes. We call these cells haploid. We call the old cells diploid. Yes?
Oh no, that's the thing with the 2N.
2N is diploid. That means 2 sets of chromosomes. And these only have 1. Any more questions right now?
What
Yes.
is that? What is that?
What's
any more?
what is the ploid in diploid? Like, why is it di? Doesn't di mean 2?
Di means 2, so 2 sets of chromosomes.
Right, okay.
Haploid is kind of half that. Question? Okay. We're not done yet. Even though we have 1 set of chromosomes, each chromosome still has sister chromatids, and we got to separate those. So, what happens is, the second set, meiosis 2 is what we call it. Again, during this, do you know what this time out phase is called? Between...
Chill time.
It has a name, it's not called chill time. Interkinesis.
Hello. Nice.
Anthony, was that you?
Yes, sir.
Good job.
Yeah, whatever.
Interkinesis is a time where the nuclear membrane dissolves. Could y'all quit doing that? It's often y'all 3.
I didn't know.
Tucker, I know, it wasn't this time, it was Tucker this time, but it's often y'all 3. Somebody makes something and then you make a side comment, and it's distracting, okay? That was great in 5th grade, 6th grade, 7th grade. It's too late for that now. You got to be mature students, respect other people, so forth, okay? Could y'all just stop doing that one? The 3 numbers? Ridiculous. Talk, second. Thank you. Okay. Where was I? Oh, the nuclear membrane dissolves. The chromosomes coil back up so you can see them. And during interkinesis, the centrioles had copied themselves again. So now they can move to opposite sides and shoot out spindle fibers. All that's prophase 1. How is this prophase 1 different from, how is this prophase 2 different from prophase 1?
No crossover. Less chromosome.
There are less chromosomes, you're right. In J,
No crossover.
they don't cross over. There's no pairing up with the homologous pair, because there is no homologous pair. There is no other big one, so there's no pairing up. Question?
In the picture, they don't, like there's still a nuclear envelope and all that kind of stuff. It's not having.
This picture,
Yeah.
it's disappearing.
Okay.
It's in the act of disappearing.
Sorry.
Okay?
That's
That's okay.
Which was, I have a question real quick.
The next one, it should be gone in metaphysics. Yes?
In prophase 1, when they like mount each other and like switch the...
Yes, crossing over.
Yeah, crossing over. Do only one of the chromosomes cross over?
No, every one of them crosses over many times. So back here, let me rewind this thing. So when they were here, when they were like this, I switched these pieces. In actuality, these things are actually laying over the top of each other. and many pieces are being switched. Okay? So that this chromosome wouldn't have just one white piece. It would have several white pieces. And it's random how it switches. So it's not the same piece is going to switch every time. Maybe this piece switches the next time, and this piece switches a different time. So that's all random. And see, that gets random change into our sperms and eggs. No two sperm will ever be the same. You can have one kid, and the next time you have a kid, it'll be different, for sure. And one of the reasons is because of this switching of pieces is random. And the lining up during metaphase is
What
random.
if you have a disease where your cells won't switch?
I've never heard of that. I don't think that exists.
We could try to
I thought it was no switchy silly disease.
Okay, so that's prophase 2. The nuclear membrane dissolves, the chromosomes coil up so you can see them. Then metaphase 2, they line up in the middle. You know, they're attached to the spindle fibers and all that. You can write about that if you want. Anaphase 2. What happens in anaphase? The sister chromatid split. It's just like mitosis. Meiosis 2 is just like mitosis. And then finally, telophase 2. A new nuclear envelope appears.
And nuclei.
New nuclear envelope. New nucleoli, yes, yes. I'm not going to draw that in, if you don't mind. What happens to the chromosomes now?
They uncoil.
They uncoil or
They decondense.
decondense, good word.
Is it chromatin?
It's called chromatin with an N on the end. Chromatin is what it's called when it's uncoiled. Just a fishing line laying out everywhere. Yes, Grant?
Is there a reason that, like, this time it was vertical instead of horizontal?
Nah, it's just easier for me to show. If I do them all horizontal, it looks confusing. It can actually happen anyway, in any orientation. Now look what we have. We have 4 cells where we started with 1. Each of them is haploid, has half the number of chromosomes. Each is different. Do you see how each is different?
Yes.
Yeah, one's got.
If this is in a testes, each of these will grow tails and become a sperm cell. In actuality, the tails are real long. This part is real small.
Have you ever been doing this for a class and accidentally want two of the same? Like in your demonstration?
They can't be the same if you do the crossing over.
I know, but like in your demonstration.
They can't be the same if I do the
Oh.
crossing over.
So they're not
That assures it being different.
So they were not them crossing over.
Have I ever forgotten to do the crossing over? No. I'm perfect.
Grant, I see the logic.
You're the pro. No, it can't be. They crossover.
Now, why do we want variation?
So they all look different.
Look at the puppies. See how all the puppies look different a little bit? If you look close enough, there's differences in all of them. And they look different from the
That
parents.
one and that
This is a little video showing you why variation is important.
Little puppies.
And they blow.
Pay attention to this. This is funny. Eventually it'll start. I hear you.
When any organism reproduces, it makes new individuals that have the same basic characteristics as itself. Offspring share with their parents a kind of body plan that remains essentially the same generation after generation. For example, this mother and her kittens have fur, 4 legs, whiskers, and other characteristics that distinguish them as cats, not fish or birds or people. But each kitten is also different from its parents and its brothers and sisters. Most noticeably, each has different color fur, but there are many other differences as well. While it is important for all members of any species to have the same basic characteristics, genetic diversity is also important. In fact, genetic diversity helps a species survive.
To see how, let's imagine a mythical species of animals, which we'll call wimpogs. Suppose all wimpogs are genetically alike in every respect, and that they all have characteristics that help them survive in a warm environment. Up to now, wimpogs seem to do fine just the way they are. But now suppose that conditions change and the environment grows colder. The same genetic characteristics that help this species survive in a warm environment might now threaten its survival. The species could become extinct. But with genetic diversity, the outcome might be different. Let's suppose now that some individuals of the Wimpog species have characteristics that would help them if the climate gets colder. If such a change in climate occurs, these individuals will be better able to survive. If they reproduce, they also will ensure that Wimpogs survive as a species.
While Wimpogs are imaginary, in reality, different species benefit from genetic diversity. Genetic diversity provides options to a species that may help it survive when conditions change.
Okay, so what was the point of that? Well, if the kids are all different, some are going to have more fur, some are going to have less fur. Some are going to have big ears, some are going to have small ears. Some are going to have big eyes, some are going to have small eyes. And by making your kids different, your species has a better chance of surviving if conditions change. If it gets real hot, the ones with less fur will survive. If it gets real cold, the ones with more fur will survive. And so species are always changing because of the differences. The changes are slow, but they happen over millions of years. And we study that. It has a name. It's called evolution. Evolution is species changing. And the reason for evolution is the differences that we see during meiosis. These differences are what cause, this variation we see is what causes evolution. And there's 3 types of sources of variation that we've studied. There are 3 sources that make kids different from the parents. Number 1, crossing over. When we change these pieces, when these pieces switch from one to the other, that's a source of variation. Number 2, independent assortment. When all the chromosomes line up, and they can line up randomly, that causes variation in the sperm cells. And the third one that causes the most differences in the kids is sex. If I'm of a family and I mix my genes with someone of a completely different family that has completely different genes, the kids will really turn out different than either of the parents. So that's the purpose of sex. If you're ever asked the question, what's the purpose of sex, you'll probably say something like, oh, it feels good. Or it's fun. Or I like to watch it on TV. None of those is the purpose of sex. The real purpose of sex is to mix the genes with someone from a completely different family, and that'll make the kids turn out totally different than anything that ever existed before. And the differences in the kids allow a species to have a better chance of surviving if conditions change. That's the purpose of sex. Yes?
Why, when people have sex with people in their family, do kids get over?
Because, that's a good question. Because there's more chance of bad genes getting together. For instance, everybody contains about 6 lethal recessive genes. And next we're going to talk about dominant and recessive, and I'll explain what that means. But lethal means deadly, and recessive means hidden. So I contain a few lethal recessive genes that if my lethal recessive genes combine with another person's lethal recessive genes, the baby will die or have bad problems. Well, if I have sex with someone in my family, have babies with someone in my family, it's very likely that we'll have the same lethal recessive genes and they'll get together. But someone from a different family probably won't have the same lethal recessive gene. So you don't want to have babies inside your family, and that's why incest is taboo. Actually, you are programmed to not really be attracted to people in your family, even though others might be. Even though they might say, oh, your sister's hot. You go, oh, no, she's not. Because you are programmed. You don't want to have babies with someone in your family. That's pre-programmed. Yes?
So, like lethal recessive genes, is that also like when a baby is retarded is born?
It could cause retardation. It could cause lots of problems. We see it in the animal kingdom a lot when groups are reduced to very small numbers, like the manatees are about to go extinct and there's only a few hundred of them left. Well, they're pretty much having to reproduce with people in their own family, and so you get a lot of these lethal recessive disorders, and they're in trouble because there's nothing else to reproduce with. That's the only thing, is they're all just a few families left. So that, yeah, that causes problems. Good question. Good thinking. Okay. So, that's that. You got to write about that if you get this essay. Next thing we're going to talk about, I'm going to start today, I'm going to finish tomorrow. I'm only going to do about 10 or 15 minutes now. Is a guy named Gregor Mendel. You're going to learn Mendelian genetics now. And when I say the words Mendelian genetics, often kids get scared. And they think it's really hard. But not that bad, and I'll show you why. Mendel was a monk. And he lived in Austria in the mid-1800s. And he lived in a monastery. And he had a garden. And he grew peas, pea plants. You see he's holding a pea plant there in the picture. These are peas, like y'all have eaten pea soup and that kind of thing. You know what peas are. Well, Mendel's famous for his pea plant experiments. And he figured out heredity. He figured out how genes are passed from parents to offspring, and why the offspring look the way they do, why they look similar to the parents. He figured it all out in the 1800s. Back then, scientists didn't communicate very well with one another. They didn't have the internet, they didn't have phones and stuff like that. So his work kind of went unnoticed. Nobody really knew what he was doing. And he had figured out how genetics works. And 20 years later, there were scientists in other places in England and Germany still working on it. And he had already figured it out 20 years before. Imagine if you worked on a problem for 20-something years, and then you picked up a book one day, and everything you were working on had already been figured out. 20 years of work. That happened to some of the scientists. Isn't that crazy? So Mendel, he figured it out. These are the pea plants he used. And pea plants, the way a plant works is a plant has, the reproductive structure of a plant is a flower. Think about that next time you're picking flowers. You're picking reproductive structures. And then you're sniffing them. Ah, it smells good, the reproductive structures. Imagine if somebody tried to do that to your reproductive structures. Pick them and sniff them. That wouldn't be too good. Anyway, here's the flower, and these are the male parts of the flower. They're called the stamens. And this is the female part of the flower. Most flowers have both male and female parts. Hold on, this is about golf. I got to get this. Tom Willis. Yes. Okay. Okay. Okay, could just call me back. Okay, thanks. Okay. All right. Yeah. So the flower, what Mendel did is he would mate these flowers with one another. He could mate the male parts with the female parts very easily. What he would do is he'd take a paintbrush, you see a paintbrush here, and he would brush the paintbrush up against the male parts of the flowers, and the male parts produce pollen. Pollen are plant sperm. Did you know that? Pollen grains are plant sperm. And he would brush a paintbrush up against the male parts, and he would take them over to the female part of another plant and brush the pollen onto the female part. Thus, he knew how to mate one plant with another. Brush the pollen off the male part of this one, bring it to the female part of that one. And then, the pollen grains, the sperm, would fertilize the eggs. These are the eggs, these little dots here. And the pollen grains kind of move down a tube and fertilize the eggs. and then about a few weeks later, the eggs grow into peas. And then he would plant the peas, and the next generation would grow from that. So you can make this one and this one, plant the peas, and then the next generation grow up. And you could see how they look. Yes?
So does every plant produce pollen for their plant?
Yes.
Okay.
Not every plant, most plants. We'll study some plants that don't do it quite like that, but almost all of them. Okay, so Mendel could make peas, and let me show you what he did in his experiment, and you see if you can figure out genetics the way he did. Mendel took, okay, hold on. Let me draw the ground. This is the ground. That's the ground. Mendel took tall plants. I'll draw a long green line means tall plants. Actually, I'll just draw one. Mendel took a tall green plant, a tall pea plant, and he mated it with a short pea plant. Now, at the time, people believed in what we call the blending theory of inheritance. The blending theory of inheritance is kind of common sense. If you made a tall pea plant with a short pea plant, what do you think you would get?
A medium.
Medium. That's blending theory. And Mendel proved blending theory wrong. Mendel mated a tall pea plant with a short pea plant. And you know what he got?
Tall.
Taller.
He got all tall people in the next generation.
Dominant gene.
Now, he called this first generation the P generation because P stood for parents. So these are the parent generations. Let me turn the light on so the camera can see this better. This first generation was called the parent generation. This next generation he called the F1 generation. F1 stands for first filial. That means first set of sons, or first set of brothers. I can't remember which it is. Anyone here took Latin? What's filial? Brother?
Family size.
Daughter?
Daughter.
Sons? Okay, I don't know. He called this the F1 generation. The first set of kids was the F1 generation. And this was the P generation. So he made it a tall and a short. He got all tall. That doesn't follow the blending theory. Mendel's already proven the blending theory wrong.
Yep.
Then Mendel said, what I'm going to do is I'm going to make two of the F1 plants together. He took two of those and he mated them together. X means mating, by the way. X, he called it a cross. He crossed this plant with that plant. That's a cross. And by the way, whenever you do a cross between two different types of plants, the new one is called a hybrid. So when you cross that one with that one, these are called hybrids.
So you're about to have a hybrid of a hybrid.
And then he crossed two of the hybrids, and guess what he got in the next generation?
Short.
Okay, tall.
Taller.
And short.
He'd get 3 tall to 1 short.
Ooh, what wrong?
He called this the F2 generation. Now Mendel looked at those results, and he said, And he looked at the other monks in the monastery, and he said, And they said, why are you saying, And he said, I've got some weird results. And they said, oh, what's for dinner? And he said, pea soup.
No, look at that. Because he just got a bunch of peas.
So he has a bunch of pea plants. So why? Can you figure out what's going on?
I can't.
Mendel was a math teacher and a science teacher as well, as a gardener. And he figured out what was going on. What's that?
The gene still stays in the
Is the recessive gene still transferred over to all the hybrids?
Yes, he figured out dominant and recessive genes were working here. And let me show you what he figured out. He figured that these first 2 plants, he figured every plant has 2 genes for a trait. Graham, hang with me. Seem like your hand's in front of your face. Every trait has 2 genes for it, but he didn't call them genes, he called them alleles. He said that height in a plant actually uses 2 alleles. And he called them, in this plant, he called them big T, big T. Big T means tall. In this plant, he called them little t, little t. Little t means short. Big T equals tall, and little t equals short. And every plant gets 2 genes. 2 genes for each trait.
That's why it's big T, little t.
Ah! When, Mendel said, when plants mate, they only donate 1 of their 2 letters to each kid.
Oh, we learned this last year.
So this plant donates the big T to this kid, and this plant donates the little t to that kid.
Shouldn't it be like medium height?
So that's big T, little t. Now shouldn't it be, who said that? Shouldn't it be medium height? Apparently not, because they all grow tall. So Mendel said the big T must dominate over the little t. And so he called T a dominant gene. Hold on, I gotta get this again. Tom Willis. Hello. No problem.
So each trait
Okay.
is 1.
No, it's not.
Gotcha. We'll do it.
Right, hello, this is Rick.
No. They'll just jump in the bus. We won't have time to wait for them to get out of the cafeteria. Tell them to jump in the bus, and I'll get them something when we get there. Okay, so the other kids can be warming up. Okay, just tell them to meet us out front as soon as they're done. Out front of the school. I'm going to pull the bus back to the front of the school after we eat. Okay, good deal. Okay, thanks. Bye. All right.
So wait, each gene gives 1 trait?
Each parent gives only 1 of its genes to each kid.
For each thing.
For each trait.
Yeah.
This one makes little squares.
Yeah, we're going to do those.
Okay.
I'm going to show you how to do those tomorrow.
Okay.
I just said that.
So, that's big T, little t. That's big T, little t. That's big T, little t. They're all big T, little t, so that's why they're all tall, because tall is dominant over short. However, if you make 2 of these together, now see if you can follow. Here's where it gets confusing. There are 4 possibilities. A, this parent could give that big T, and this parent could give that big T, and this kid would be big T, big T. And that's a tall kid. This parent could give that big T, and this parent could give that little t, and it's a big T, little t. Or this parent could give this little t, I'm sorry, this parent could give this little t, and this parent could give this big T, and that's little t, big T, but you always put the uppercase letter first. So, you always write it big T, little t.